children, you have seen the chapter, the principles of inheritance and variation, till Mendelian principles you had seen. Today we will deal with the topic chromosomal theory of inheritance. But before starting chromosomal theory of inheritance, we should know what is a chromosome. Chromosomes, as the name suggests, chromosomes, that is colored bodies. Chromo means colored and somes means somatic bodies. Colored bodies or colored somatic bodies are known as chromosomes. Now, you had seen in your earlier classes, like in 11th we had dealt with the chapter cell cycle and cell division, in which you had seen how the chromosomes are dividing and how the chromosomes are separating. Starting from that, if we see, this is the cell cycle. If we see the cell, the cycle is divided into 3 phases you have seen: G1 phase, S phase, G2 phase, the 3, and the mitotic phase. Here a short phase is there, that is G0 phase, that is known as the quiescent stage. Quiescent stage means the cell here does not divide, it takes a little rest. Quiescent stage can also be known as the resting stage. It can also be known as the resting stage, quiescent stage, G0 stage. After that, then after the G0 stage, the cell will undergo the G1 phase, that the cell will grow, it will duplicate itself, all the things, means it will assemble all the things, there will be ribosomes, endoplasmic reticulum, which are required for protein synthesis and other functions of the cell. Now, next phase you see is the DNA replication. Cell growth you have seen, cell growth, G1 phase, it takes place about 10 hours. At the G1 phase, that is the growth of the cell, it takes place about 10 hours for the cell to grow. Then next phase is the S phase, that is the synthesis phase, in which the DNA replicates. DNA will replicate and it will form a complete replica of its own, that is replication of DNA will take place at this, and it is of 8 hours. Next stage is the cell growth or the G2 phase. Here also cell growth takes place, it is about 4 hours. Then after all the assembly, after taking rest, after assembling things, after DNA replication, again after cell growth, the cell undergoes the M phase. It is nearly about 1 to 2 hours. M phase is of 1 to 2 hours and it is known as the mitotic phase or the meiotic phase. It is the M phase. In the M phase, it is divided into 5 substages, that is prophase, metaphase, anaphase, telophase and cytokinesis. If these all the substages are similar in meiosis also and in mitosis also, all the substages are there in mitosis as well as in meiosis. But if it is an M phase, that is this M phase is a meiotic phase, then in the meiotic phase we know the meiosis is divided into 2 substages, meiosis 1 and meiosis 2. Of these 2 substages, the prophase, meiosis 1, in meiosis 1 the prophase is of a very larger duration, prophase is of much larger duration in meiosis 1. Why the prophase is of much larger duration in the meiosis 1 is that prophase is divided into 5 substages. The prophase of meiosis 1, meiosis 1, prophase 1. Prophase 1 is divided into 5 substages: leptotene, zygotene, pachytene, diplotene and diakinesis. If we see in the prophase 1, we know that suppose this is the nucleus, we know whether it is mitosis or meiosis. Meiosis particularly, it takes place meiotic division. Meiosis will take place inside the cell. Why inside the cell? Because inside the cell chromosomes are there. See, chromosomes, when it is inside the nucleus, if this is the cell and I am considering the cell nucleus, that is nucleus of the cell, this is the nucleus of the cell, and inside the nucleus we are seeing that here tangled bodies are there. these tangled bodies which are there, this is also known as chromatin. It is known as chromatin, or it is known as chromatin thread. We are seeing that the chromatin thread, it is tangled. See, it is tangled. We are just seeing it is a thread, like a bunch of thread. Okay. So that's why in prophase 1, meiosis 1, prophase 1, it is divided into 5 substages. Why? Because prophase is of much longer duration in meiosis 1. This tangled parts, if we see first in the leptosene stage, this part only I am showing here inside the cell. All the things which I am showing here, this is not happening outside the nucleus, it is all the stages will be happening inside the nucleus. So in the first stage leptosene, what will happen? These threads, they will separate, chromatin threads, it will separate, it will condense and it will form short threads. Next, in the zygotene stage, what will happen? So these threads, these chromatin threads, when these are separated, these have condensed, now this will be known as chromatid. Chromatid. Now it will be known as chromatid. Now when they will come and lie side by side, see there are 2 types of chromosomes we know, that is homologous chromosomes and heterologous chromosome. 2 types of chromosomes, homologous chromosomes, heterologous chromosomes. So when the pairing will take place in the next stage, this is the leptosene stage, in the next stage zygotene stage, that is the pairing will take place. So when the pairing will take place, these 2 will come and lie side by side, that is the homologous chromosomes will come and lie side by side. Everywhere this pairing will take place during the zygotene stage. Next stage, in the packaging stage, what will happen? Crossing over takes place between non-homologous chromosomes. Homologous, I have written heterologous, can be called as heterologous chromosome as non-homologous chromosomes? See, there are different names for it. Homologous chromosomes, non-homologous chromosomes, homologous chromosomes or the sister chromosomes, non-homologous chromosomes, non-sister chromatids or non-sister chromosomes. So what is happening in the homologous chromosomes here in the zygotene stage, they will come and lie side by side, that is pairing takes place. Now in the next stage, packaging stage, crossing over will take place between the non-homologous chromosomes, means between this chromosome and this chromosome. So suppose this is one chromosome and this is one chromosome, just for a differentiation I am marking it as this way. It is suppose this one, this. So this is one homologous chromosome, this is one homologous chromosome. So pairing will occur between this or this. Any one can pair. So any of the chromatid, means a pair of chromatids together, when they come together and they are joined by a centromere. When they are joined by a centromere, now they will be known as chromosome. Unless and until they are not joined by a centromere, they will be known as chromatids. When the chromatids come together and they are joined by a centromere, now it will be called as a chromosome. So the crossing over will take place between non-homologous chromosome, that is the packaging stage. Then in the diplosene stage, what happens? When the crossing over takes place, crossing over occurs at particular points. We have already seen that in class 11 during your cell cycle and cell division. You have already seen this, I am just repeating it. So when the in the packaging stage, what happens? There are specific points where the crossing over takes place. Those points are known as chiasmata. Those points where crossing over take place, those points are known as chiasmata. So when after the crossing over takes place, after the crossing over, these separate. It is during the diplotene stage, the crossed over parts will separate. See here one part, suppose here one part, crossed over parts will separate along the chiasmata, means terminalization will take place. So terminalization occurs in diplotene stage, and diakinesis, now they will be separated along with the crossed over parts in the diakinesis stage, and they are joined by the centromere again. So that's why the prophase 1 of meiosis 1 is of a very longer duration. So how is it related? Why it is related to chromosomal theory of inheritance? Our topic for today is chromosomal theory of inheritance. Our topic for today, it is chromosomal theory of inheritance. I had told you all the things how the chromosomes are behaving. Now we will see what and how there is a comparison. Chromosomal theory of inheritance means it is a comparison between Mendelian factors and the chromosomes. Sutton and Boveri, the two scientists, Sutton and Boveri, they gave chromosomal theory of inheritance in 1902. So what does chromosomal theory of inheritance deals with? We'll see just now. Children, we had seen like how Sutton and Boveri, they gave the chromosomal theory of inheritance in 1902. Sutton and Boveri in 1902. We knew like it was Mendel who basically he discovered chromosomes, but that time he was not able to tell it as chromosome. He called them as factors. Even genes were not known by him. That's why he called them as factors, alleles. These all were the names in spite of chromosome and genes. That was being given by Mendel. Mendel, because his concept was very, at that time the scientists did not accept his. He gave it in 1865. After 35 years, his findings were proved by Sutton and Boveri. That is in 1902. That's but why? Because first of all, Mendel could not explain what are those factors. That's why it was not accepted. Next, he used biology along with mathematics, that is genetics, biology along with mathematics. And at that time, scientists was not able to comprehend how we can join mathematics and biology together and how can we come to a conclusion. Hence, his theories was not accepted at those times. But he was the base. He provided a base to what we know today about chromosomes, genes and all. It was being provided by Mendel, and it was Sutton and Boveri who formed a relationship that what Mendel gave and what we are following today, that is the chromosomal theory of inheritance. Chromosomal theory of inheritance. They formed a parallelism between Mendel and their theory. They formed a parallelism between Mendel's theory and their own theory. First we will see basically what is the difference between mitosis and meiosis. Why mitosis is called equational division and why meiosis is called as reduction division. This I have drawn about mitosis, this whole thing. This is mitosis. The division in mitosis as equational division, and this I have drawn is meiosis. The division in meiosis is known as reduction division. So we'll start from the very starting. Basic means what is mitosis. See, if you see in this cell, the chromatids are there. Yes, they are here. The centromere are there. Here also you are seeing chromosomes. They are separated. When the chromatids, the chromosomes, when they are in the which stage? That is the S phase. That is DNA replication will take place in the S phase. That is the synthetic phase when the DNA will replicate itself. See here only one strand of chromosome. So chromosome doubling takes place. First we will see mitosis. When chromosome doubling takes place, see it has formed its own replica. Here one chromatid, here a chromatid along with one more chromatid joined by a centromere. Hence it is called a chromosome. A pair of chromatids is known as a centromere chromosomes. So here also you are seeing, see, single. Here also it is formed its replica. Here it has replicated. Here 1 point. Here also it has replicated. So this is when cell division, when the cell division will take place in mitosis, when the cell will divide in mitosis. Cell divides into two. This cell will divide into two. If you observe carefully, you will see this one strand, it is going here. Here one strand, it is going here. This one strand, it is going here. This one strand, this cell, this here, this. Exactly equally it has divided. Now if you see again, see it mitotic division first it is 2, then 4. These two cells will again undergo chromosomal doubling. That means it will again undergo the synthesis phase. The synthesis phase will again go, the DNA will again replicate itself. So here again see, this is one strand, it has replicated and it will form the other strand. Similarly see 1, 2, same, you are seeing DNA has replicated. Now this, after this again the cell will go for cell division. Again when the cell will go for cell division, you are seeing, see, equally it is divided into 2. Here also one strand, here one strand, here one strand of this strand, here one strand, here one strand. Here this chromosome, if you are seeing, this one strand here, this one strand here, means they are equally dividing right from the very beginning, starting what the cell had. See what the cell had, same thing is retained. Each cell is having the same number of chromosomes in mitotic division. See, we started with this cell, we are ending with that cell, each cell is having the same chromosome number. Okay, so what it means the cell is dividing equally. Each cell retains the normal chromosome number in mitotic division, whereas when we see the meiotic division, and hence that's why the mitotic division is known as equational division, equal. We started with this many chromosomes, we are ending with these many chromosomes in each cell. So when we see in meiosis, if you see in this cell, again it will also go, DNA replication will take place in this also, synthetic phase, DNA replication, the cell will go, see, it will replicate, one, it has formed its copy. Here this cell, this chromosome also, it has formed its copy, it has replicated in its form copy. Here one strand, here two strand. Similarly here you are seeing the chromosomal doubling is taken place. Now after chromosomal doubling, what will happen? As we said, meiotic 1 is a larger stage. Prophase 1 of meiotic 1 is a larger substages. It is divided into 5 substages: prophase, then the leptotene, zygotene, pachytene, diplotin. It is in the pachytene stage that pairing takes place. It is that stage. So here the chromosomal pairing is taking place in the pachytene stage. Pairing will take place, the crossing over will take place till diplotin stage. It will be there, the crossed over parts will be till in the diplotin stage. At the end of the diplotin stage, means diplotin stage is ending and dikinesis is starting, the crossed over parts will separate, that is terminalization will take place. The same thing which happened in 11th standard cell division chapter. So here the chromosomal pairing is taking place, that means here meiotic 1 is taking place, meiosis 1. Meiotic 1 is, meiosis is divided into 2 substages, meiosis 1 and meiosis 2. But mitosis is not divided into any substages, it is only mitosis. So that's why meiosis 1 is known as reduction division, and meiosis 2 is known as equational division. Or we can also call the equational division as mitotic division, or the mitosis is taking place in meiosis 2. So what is happening here? Chromosomal pairing is taking place. So here if you see, all these things are coming and lying together. They are coming and lying together means pairing is taking place, and after that then the cell division takes place. When the cell division takes place, if you see, this full half reduced, one full set is going in one cell, one full set is going in another cell. This one set is going in one cell, this one set is going in one cell. Here what happened in mitotic? They divided equally. See, this was one set. Of the one set, one went in one cell, one went in one cell. But here the complete set is going in one cell, and this complete is going in one cell. That means equational division has not taken place. That is, it has reduced. Half is going in one cell and half is going in the other cell. Here only we know that why meiosis is known as the reduction division. Why? This is the reason why meiosis is known as the reduction division. After that again it will go to meiosis 2. Meiosis 2 is what? Meiosis 2 is just like mitotic division. So that's why again it will undergo cell division, that is meiosis 2. Then you see how it is equally dividing. Here there are 4, that is the chromosome, a pair of chromosome of which one chromatid goes in one cell, of Of this, one chromatid goes in other cell. This one, it is going in this cell, this it is going in this cell. Here also see, it is dividing equally. This one chromatid is going in one cell, one chromatid is going in one cell. This one chromatid in one cell, this one chromatid in one cell. So this part where this cell division took place in the second time, this cell division is meiotic 2. Meiotic 2, meiosis 2, meiotic 2, or it can also be known as mitotic division. And this which took place here, the cell division which took place here after chromosomal pairing here which took place, this one, this is meiotic 1, this is meiotic 2, this one is meiotic 2, this one is meiotic 1, this part, till this part it is meiotic 1. After this part which has taken place, that is cell division, this is cell division 2, that is meiotic 2. Written it here, it should be written here. That's why I put the arrow here, just for no confusion. So this is meiotic 2, you are seeing equally it has divided. This is the equal division, which is, this is the half number of chromosome. Each sperm has got half the number of chromosome. That's why meiotic 2, it is after meiosis we are getting 4, 1, 2, 3, 4, and it is half the number. But whereas in mitotic it will continue to go 2, 4, 6, these 4 cells again it will undergo mitotic division, this will give rise to 2, this give rise to 2, so 4, then here 4, it is 8, but here you are seeing 1, 2, 3, 4, 4 sperms are found with which are having the normal number, but it has half the normal number. Here you are seeing, till here you are seeing that it has got the same number. From here when it is taking meiosis, meiotic 2 is happening, half the chromosomes are going. That's why meiotic 2, the chromosome numbers become half, and in meiosis 1 the number retains what the original cell had the number, same each cell will have the same chromosome number, that is chromosome number retains in mitotic divisions. That's why it is known as equational division, equally divide. Whereas in meiosis it is known as reduction division, because after chromosomal pairing the first meiotic division which takes place, here you are seeing it becomes half. 2, see, this one complete cell is going in one cell, this complete set of chromosome is going in another cell. That means each cell are not having the equal parts. One is having the complete one part, other cell is having the complete other part. That is, it has reduced. One cell is getting this one, but none of the portion of this chromosome is entering this cell. That means it has reduced. Meiosis 1 has reduced, that this is reduction division, and meiotic 2 is, again you can see this here there were 2 each, see in here also one part went and here also one chromatid went, that is it has divided equally, and hence this is the difference. This was given by Sutton and Boveri. Sutton and Boveri said this only, what Mendel had told. Mendel had told, you had already, it has been discussed in your previous chapter, what Mendel told, that the gametes segregate, first they occur in pairs. What Mendel told, that factors occur in pairs. The pairs, they were known as the allelic pairs, they will occur in pairs. Similarly, like see, the chromosome are also occurring in pairs, first point. Then according to Mendel, second point, what he told, that the gametes will segregate, means the chromosomes will segregate at the time of gamete formation. Mendel told factors segregate at the time of gamete formation. Here also you are seeing segregation has taking place, here also segregation is taking place. So this was what Mendel want to say, but at that time there was no such microscope, the chromosomes were not formed, and that's why he termed those as factors. Those factors were nothing but these were genes, and that was being proved by Sutton and Boveri, that yes, it is the chromosomes that are separating, they occur in pairs, and the genes which are there, they occur on the chromosome. That is the chromosomal theory of inheritance, which was proved by mitosis, meiosis, as well as it was related to Mendel's theory, and then a parallelism was formed between Mendel and Sutton and Boveri, and hence it was proved that yes, it was nothing but the factors are nothing but these are chromosomes, and hence the chromosomal theory of inheritance means it is the chromosomes what we are inheriting from one generation to the another generation. These chromosomes are being passed on from one generation to the other generation. We will now see the sex determination, how the sex determination takes place, how the sex of the organism is being determined. We know that there are two sets of chromosomes, so and that it is X and Y, what we know today. But in some organisms what happens, there are XO type of sex determination in exotype, and in some like domestic fowl it is ZW. Here in insects we are seeing this, we are seeing in insects, here two types of, in the this sex is determined, it is determined by the male in the insects. Why? The female has got 2 X chromosomes and the male insect has got 1 X chromosome and another is null or void, means non-functional. That means only 1 X will come from the male. So suppose if the sex has been determined, the sex will be determined by the male counterpart. When fusion takes place, suppose here, if this fusion takes place, here this male and female, this, it will be the female insect, here again it will be the female. If the O mates with the X, then it is a male, then it is a male. That means the determination power or the sex determination power is with the male. That's why it is the male will determine the sex of the organism. It takes place in insects. Similarly, if we see in birds and domestic fowls, it is this type of formation is being seen. This is the male, they are having the same type of gametes apart from the autosomes, the sex chromosome, it is 2Z, and in the female it is ZW, and hence in this if you see, the male has got 2 Z chromosomes, in the sex chromosome that has 2 Z, but in the female one is Z and one is W. Again means the sex will be determined by the female counterpart, that is in domestic fowls and all. So here you are seeing in insects the male is determining the sex of the organism, whatever the progeny, the sex of the progeny will be decided by the male counterpart, whereas in the domestic fowl the sex will be decided by the female. It is the female which is heterogamete, means 2 different type of gamete are present in the female. So she will decide what will be the sex of the progeny. Similarly in humans we are having XX and XY. This XX gametes, it is in the female, human female, and XY is in the human male. That's why the sex of the progeny will be decided by the human male, because it is heterogamous, 2 different type of gametes are present. Whereas the female, they don't have a deciding factor because both the gametes are similar gametes. That is the meaning of sex determination, means which person or which organism will decide whether it will be the male organism or the female organism, depends on the organism itself. Like in insects, it is the male or insect that will decide the sex of the progeny. In birds and fowls, it is the female which will decide the sex of the progeny. In humans, it is the male which will decide the sex of the progeny because they are heterogametes. Like suppose if they are going undergo fusion will takes place, here XX, that is the female child, here XY, this is the male child, then again XX female child, XY male child. Here we are seeing we are getting 2 X similar types of gametes, and hence it is a female. Here 2 dissimilar type of gamete, and hence it is a male. That means it is the male which is deciding what will be the sex of the progeny, and hence this is how sex should be determined. So I suppose that you have understood about everything what I had taught today, chromosomal theory of inheritance, the relation between Sutton and Bovary chromosomes and the Mendelian factors, and then how the sex should be determined in insects, in fowls and in humans. Next we will continue with recombination and linkage, that is linkage and recombination in the next class.